Pinch yourself right now, your forearm, the back of your hand, anywhere. Just a quick, sharp squeeze. Feel that? That sting, that brief flare of hey, stop that. You felt it because a cascade of electrochemical signals just fired from the nerve endings in your skin, raced up through sodium channels embedded in your pain-sensing neurons, hit your spinal cord, climbed into your brain, and within roughly 150 milliseconds, registered as something unpleasant. The whole system worked perfectly. But here's the thing. If I could somehow line you up next to 100 other people from 100 different genetic backgrounds and pinch every single one of you with exactly the same force, in exactly the same spot, you would not all feel the same thing. Not even close. Some of you would barely flinch. Some would wince hard. And the genetic reasons for that gap, the actual DNA variants that determine who suffers more and who shrugs it off, are so unevenly distributed across human populations that, when researchers finally mapped them globally, the patterns defied every assumption the medical community had been operating under for decades. Your pain is not just in your head. It's in your genome. And your genome didn't come from nowhere. It came from a specific lineage, a specific migration path, a specific set of ancient pressures that shape the molecular machinery sitting inside your nerve cells right now. So let's talk about what that machinery actually looks like, who got what version of it, and why the differences between populations are so much larger than anyone expected. For most of recorded medical history, pain was treated as a universal constant. You stub your toe, you feel pain. The stimulus is the same, so the experience should be the same. Variations in how people reported pain were chalked up to psychology, culture, stoicism, exaggeration, anything except biology. Doctors in the 19th and early 20th centuries routinely ascribed pain sensitivity differences between groups to willpower or temperament. It was patronizing, it was wrong, and it held back real science for generations. Even into the late 1990s, the prevailing view in clinical medicine was that pain thresholds were essentially uniform across healthy humans, and that differences in pain reporting reflected differences in coping, expectations, or social conditioning. Twin studies had begun to hint otherwise. Norbury and colleagues at King's College London published data in 2007, in Brain showing that heritability of pain sensitivity in women ranged from 22% to 55%, depending on the modality tested. Heat pain, pressure pain, cold pain, all had significant genetic components. A broader review by Nielsen and colleagues estimated heritability for various chronic pain conditions at 50% for migraine, and 52% to 68% for lower back pain. These were not small numbers, but even then the specific genes were barely known, and nobody had systematically compared their distribution across global populations. That changed fast. And the picture that emerged was, okay, I need to be honest here, it was genuinely confusing at first. I spent weeks trying to make the data tell a simple story, and it refused. Pain genetics is not one gene, one effect. It's a layered, interacting, sometimes contradictory web of variants across multiple systems. But there are key players, and those key players do vary dramatically across populations, so let's walk through them. The most important single gene in human pain perception is almost certainly SCN9. It sits on chromosome 2 and encodes a protein called NAV1. 7, a voltage-gated sodium channel that's densely expressed in the nociceptive neurons of your dorsal root ganglia. These are the cells that detect damaging stimuli and fire the signals your brain interprets as pain. NAV1. 7 is the amplifier. It takes subthreshold electrical signals in your pain neurons and boosts them until they fire a full action potential. Without functional NAV1, 7, you literally cannot feel pain. We know this because of rare families, predominantly identified in Pakistan and reported by Cox and colleagues at Cambridge in 2006 in Nature, who carry loss-of-function mutations in SCN9A. These individuals are completely unable to feel pain from birth. They have normal touch sensation, normal temperature detection, but zero pain. Children in these families walk on broken bones without noticing. They chew through their own tongues. On the opposite end, gain-of-function mutations in the same gene cause erythromelalgia, episodes of excruciating burning pain triggered by minor warmth. Same gene, different mutation, opposite effect. SCN9A is the volume knob for pain, and it has a wide range. Now those extreme mutations are vanishingly rare. But there's a common variant, Rs6,746,030, a single nucleotide change that swaps an arginine for a tryptophan at position 1150 of the NAV1. 7 protein. It's called R1150W, and in 2010, Riemann and colleagues at the University of Cambridge published a study in PNAS showing that the minor allele, the allele, occurs at a frequency of roughly 10% in European populations and is associated with significantly increased pain perception. People carrying 1 or 2 copies of this allele report more pain from the same stimulus. 10% doesn't sound like much, but allele frequencies vary across populations, and this is where it gets wild. The R1150W story intersects with one of the most remarkable findings in recent pain genetics, Neanderthal introgression. In 2020, Hugo Zeberg and Svante Pääbo, the latter being the Nobel Prize winner who sequenced the Neanderthal genome, published a study in Current Biology identifying 3 amino acid changes in NAV1, 7M932L, V991L, and D1908G, that came from Neanderthals. These variants entered the modern human gene pool through ancient interbreeding, and they alter NAV1. 7 function in ways that increase neuronal excitability, effectively making pain neurons fire more readily. In UK biobank data, carriers of all 3 Neanderthal variants reported significantly more pain. But here's the distribution twist. These Neanderthal NAV1, 7 variants are essentially absent in people of purely European ancestry today. They're absent in sub-Saharan Africans who never interbred with Neanderthals. But they're common, strikingly common, in Indigenous Americans. A 2023 study by Hikari and colleagues published in Communications Biology examined 7,594 Latin Americans and found that on Native American chromosome segments, the D1908G variant reaches a frequency of 68%. The V991L and M932L variants reach 29%. In Peruvians, overall frequencies hit 42, 3% for D1908G. These are not rare variants in these populations. They're common, and the researchers confirmed experimentally using quantitative sensory testing on 1,623 Colombian volunteers that carrying more Neanderthal NAV1. 7 alleles is significantly associated with lower mechanical pain thresholds after sensitization, meaning more pain from the same physical stimulus, with evidence of additive effects across the 3 variants. If you're thinking about the implications of this, you should be. Indigenous Americans carry, at high frequency, Neanderthal-derived pain-amplifying variants that are virtually absent in modern Europeans and completely absent in Africans. That's not a subtle distributional difference. That's a gulf, and it almost certainly influences real-world pain experiences in ways that clinicians, who largely don't genotype their patients, are not accounting for. Now let's move to a different system. The COMT gene, sitting on chromosome 22, encodes catechol-O-methyltransferase, an enzyme that breaks down catecholamines like dopamine, norepinephrine, and epinephrine. These neurotransmitters are deeply involved in how your brain modulates pain. When COMT activity is high, catecholamines are cleared efficiently, and pain sensitivity tends to be lower. When COMT activity is low, catecholamines accumulate, mu-opioid receptor expression shifts, adrenergic receptor signaling increases, and pain sensitivity goes up. The most studied COMT variant is Val158-MET, Rs4680. The MET allele produces an enzyme with about 25% of the activity of the Val version. But the story goes deeper than one SNP. In 2005, Dyachenko and colleagues at the University of North Carolina identified 3 COMT haplotypes, combinations of 4 SNPs, Rs6269, Rs4633, Rs4818, Rs4680, that they designated as low pain sensitivity, average pain sensitivity, and high pain sensitivity. The LPS haplotype is associated with efficient catecholamine clearance and reduced pain. The HPS haplotype does the opposite. And in 2021, Korczeniowska and colleagues at Rutgers published an analysis of these haplotype frequencies across 13 global populations using 1,000 genomes data. The LPS haplotype, The one that protects against pain varies in frequency from 13% to 44% depending on population. Let me say that again. The genetic variant most clearly associated with lower pain sensitivity in the COMT system ranges from 13% in some populations to 44% in others. That's a threefold difference in the frequency of a pain-protective haplotype. And the underlying SNP driving this, RS6269, shows even more extreme variation. The VAL allele of the famous VAL 158 MET, the high-activity, lower-pain version, has a frequency of approximately 70% in East Asians, around 50% in Europeans, and varies considerably in African populations. The distribution isn't random. It reflects different selective pressures acting on catecholamine metabolism across human evolutionary history. If you're still with me at this point, and if you are, genuinely, consider sharing this with someone who might find it as gripping as I do. Then let's stack another gene on top. OPRM1. OPRM1 encodes the mu-opioid receptor. This is the primary target of your endogenous painkillers, endorphins and enkephalins, as well as exogenous opioids like morphine and fentanyl. The most studied variant is A118G, RS1,799,971, which changes asparagine to aspartic acid at residue 40. The G allele alters receptor binding, affects endorphin signaling, and has been associated in some studies with altered pain sensitivity and opioid requirements. The population distribution of the G allele is dramatically uneven. In East Asians, the G allele frequency runs between 40% and 50%. In Europeans, it's roughly 12% to 20%. In Hispanics, around 20% to 28%. And in African Americans, it drops to 15% to 74%. A study by Hasty and colleagues at the University of Florida, published in the Journal of Pain in 2012, tested 247 healthy adults across 3 ethnic groups using thermal, pressure, ischemic, and cold pressor pain. They found a significant ethnicity by OPRM1 genotype interaction. Among non-Hispanic whites, the G allele was associated with decreased pain sensitivity. People carrying it reported less pain. But among Hispanics, the trend reversed. The G allele was associated with increased pain sensitivity. Among the small number of African Americans carrying the G allele, a similar reverse trend appeared. Wait, seriously. The same allele doing opposite things in different populations. I had to reread that paper 3 times, but it's consistent with what other researchers have found. The A118G variant likely doesn't act in isolation. It's embedded in different haplotypic backgrounds in different populations. It interacts with COMT and other genes differently depending on the genetic context, and its functional effects appear to be population-specific. This matters enormously for medicine. Opioid dosing guidelines are not stratified by OPRM1 genotype. They're not stratified by COMT haplotype, and they're certainly not stratified by the Neanderthal NAV1. 7 variants that 42% of Peruvians carry and 0% of Europeans do. The clinical system treats pain as if the underlying molecular machinery is identical in everyone. The genetics says it is. And we haven't even talked about TRPV1. This gene encodes the capsaicin receptor, the protein that detects burning heat and gives chili peppers their sting. A variant called I585V, RS8065080, alters the receptor's sensitivity. People carrying the valine substitution show altered capsaicin responses, and in some studies, protection against heat hyperalgesia. In a Japanese study, this variant significantly increased capsaicin sensitivity. Frequency data from the 1000 Genomes Project shows that the distribution of TRPV1 functional variants is not uniform. Some populations carry higher frequencies of sensitivity-altering alleles. There's also the GCH1 gene, encoding GTP cyclohydrolase 1, the rate-limiting enzyme for tetrahydrobiopterin synthesis, which feeds into nitric oxide and serotonin pathways involved in pain processing. A specific haplotype of GCH1, identified by Tejder and colleagues at Goethe University Frankfurt, and published in Nature Medicine in 2006, has been shown to reduce pain after discectomy, decrease experimental pain sensitivity, and lower pain reporting in cancer patients. The frequency of this pain-protective haplotype is approximately 15. 4% in European populations, and 9% in Yoruban Africans, a smaller gap than COMT, but still a genuine distributional difference that influences who, on average, is genetically predisposed to feel more or less pain. Here's a fun fact that puts all of this in a slightly bizarre light. You know those people, maybe you are one, who really can't handle spicy food, or who flinch at a paper cut while the person next to them barely registers it. There's a single person in Scotland, Jo Cameron, who underwent surgery on her hand in 2013 and reported zero pain afterward. Doctors were baffled. She'd gone through her entire life barely noticing injuries, cuts, burns, fractures. Researchers at University College London investigated, and in 2019 published their findings in the British Journal of Anesthesia. Cameron carried a microdeletion in a previously uncharacterized pseudogene they named FAAH-OUT, combined with a mutation in the FAAH gene itself. FAAH encodes fatty acid amide hydrolase, an enzyme that breaks down anandamide, an endogenous cannabinoid. Cameron's mutations essentially left her swimming in anandamide at all times. She felt almost no pain, experienced minimal anxiety, and healed faster than normal from wounds. One person, two mutations, an entirely different experience of physical existence. The researchers noted that the FAAH-OUT deletion had never been seen in any other individual in any database. That's one extreme. At the other extreme are the Neanderthal NAV1. 7 carriers in Peru, walking around with a pain amplifier dialed up by ancient introgression. And between those poles, 7 billion people carrying their own unique combinations of COMT haplotypes, OPRM1 variants, SCN9A polymorphisms, GCH1 configurations, and TRPV1 alleles, each combination producing a subtly different experience of what it feels like to be hurt. So what does all of this tell us about human migration and diversity? More than you'd think. The distribution of pain genes maps onto deep evolutionary history. The Neanderthal NAV1. 7 variants track the interbreeding events of 50,000 years ago and the founder effects of the first Americans crossing Beringia. The COMT Val-158 MET frequency differences between East Asians and Europeans reflect tens of thousands of years of separate selective pressures on catecholamine metabolism, probably driven by differences in stress environments, diet, and disease ecology. The OPRM1 allele distribution mirrors ancient population splits and the different endogenous opioid system tunings that evolved independently in Africa, Europe, and Asia. Pain isn't just a sensation, it's a record. A molecular archive of where your ancestors went, what they endured, what killed them, and what kept them alive. Every pain gene variant in your body was selected or drifted to its current frequency because of choices your lineage made, or had made for them by climate, disease, or geography across thousands of generations. And the gap between populations, the gap the title of this video promised you, is real. It's not cultural. It's not imagined. It's inscribed in sodium channels and opioid receptors and enzyme kinetics. But it doesn't mean what you might fear it means. It doesn't rank anyone. Pain is not a hierarchy. Having more Neanderthal NAV1, 7 variants doesn't make you weaker. Having a high-frequency LPS, COMT haplotype doesn't make you tougher. These are different molecular settings, tuned by different evolutionary histories, producing different physiological experiences of the same physical world. The real scandal isn't that the gap exists. It's that medicine hasn't caught up. Pain management is still largely one-size-fits-all. Opioid prescriptions don't come with